On behalf of the Department of Medical Genetics and Genomics, I warmly welcome you all to our 2026 Student-Led Rare Disease Day Symposium. The event has been organized by our MSc cohort, and I would like to take a moment to acknowledge all of the members who have helped in today's event. A warm welcome to all of our speakers. We look forward to learn from your insights today. Before we begin, a few points to note. To view the full program, please make sure to scan and have a look at our list of speakers today. The feedback survey will again be shared at the end of the session. Upon completion, you will receive a certificate of attendance. Please remember to scan and fill. We kindly request all our speakers, staff, alumni, and students to remain until the end for a group photograph. This is our MSc family, and we are proud of it. Today is about awareness, collaboration, and ensuring that rarity never means invisibility. And now, it is my privilege to invite Professor Edward Tobias, Professor of Genetic Medicine, to share a few words. Professor, over to you.
Thank you, Deshari. Thank you. Can you hear me okay? Great.
Yes.
Well, firstly, congratulations to you and to all of our MSc student team and Dr. Sida Bhatti, who have worked so hard and who have so successfully put the program together for this Rare Disease Day Symposium today. I have the honor of being the clinical director of our master's program, having taken over that role from an inspiring colleague and friend of mine, Dr. Douglas Wilcox. It was Douglas, who set up the MSc 42 years ago, at the suggestion of Professor Malcolm Ferguson-Smith, who hoped that it would attract students from around the world. It certainly has done that, and we know that well over 1,000 students have come to study with us from many countries around the world. It has been further developed and updated, especially by my colleagues, program directors, Drs. Maria Jackson, Leah Martin, and Kirsten Mackay-Bounford, by myself and by others in our team. In 2016, I had the privilege of being able to add to the existing excellent MSc in medical genetics an additional course covering clinical genomics, some bioinformatics, and a series of computer-based genomics workshops. And the program was then renamed MSc in Medical Genetics and Genomics. Our alumni now run laboratories and departments in many countries around the world. As many of you will know, Professor Ferguson-Smith very sadly passed away just 3 weeks ago on the 4th of February. He was 94 years old. This is a photograph of him taken at the University of Glasgow in around 2004 and shared recently by Douglas Wilcox. In Glasgow, the clinical genetics department in which I work was created as a result of Malcolm's leadership. During his remarkable career, Malcolm became one of the founders of the specialty of clinical or medical genetics. He was appointed Professor of Medical Genetics at this university in 1973 and was the NHS Director of the West of Scotland Regional Genetics Service that was based at that time at York Hill Hospital in Glasgow. It was his vision and drive that led to the establishment at York Hill in 1981 of the purpose-built Duncan Guthrie Institute of Medical Genetics, where I trained in the specialty. He had an outstanding research career, resulting in many internationally important research papers. They covered a range of areas, and he greatly advanced diagnostics, genetic counselling, gene mapping, and cytogenetics, contributing in many ways to the understanding of rare diseases. He was instrumental in establishing amniocentesis, chromosome banding, laboratory workflows, and training programs. He was a brilliant pioneer whose work fundamentally advanced human cytogenetics and genome mapping. He moved to England and became chair of pathology at the University of Cambridge in 1987 and became an emeritus fellow at Cambridge Veterinary School upon his retirement in 1998. Within 4 years, he had established the Cambridge Resource Centre for Comparative Genomics. Malcolm was one of the first to apply molecular cytogenetic techniques across species using chromosome painting to reveal conserved synteny and evolutionary rearrangements between humans and other mammals. I personally was fortunate enough to have worked with both Malcolm and with his successor, Professor Michael Connor, on the genetics textbook, Essential Medical Genetics. And Malcolm was always able to put me in touch with a world expert on any aspect of the field to answer any questions I had. For example, I remember we both discussed the technical aspects of X chromosome inactivation with the person who'd actually discovered it, Mary Lyons. Malcolm spent time at Johns Hopkins University Hospital in Baltimore, USA, establishing one of the first human chromosome diagnostic laboratories in the United States and working with another world-renowned pioneer in medical genetics, Victor McKusick, who of course created the database that is now known as OMIM, or Online Mendelian Inheritance in Man. It is used by geneticists all over the world. My immediate predecessor at Glasgow University, Professor Mike Connor, also worked at Johns Hopkins. Providing some continuity in that respect, it has turned out that through my role as chair of the Education Committee of the International Human Genome Organization, or HUGO, I too now have the privilege of working together with another lead geneticist at Johns Hopkins, albeit online. That is Professor Dr. Ada Hamosh, who is the hugely inspirational current scientific director of OMIM and who has greatly expanded that incredibly helpful resource. Beyond his outstanding research contributions, Professor Ferguson Smith was an academic leader who greatly strengthened genetics as a discipline in the United Kingdom and internationally. I know that Malcolm is fondly remembered as a mentor, colleague, and friend to so many past and present members of the clinical genetics department in Glasgow and elsewhere, and he has left an incredible legacy in the field of genetics. I personally owe a great deal to Professor Ferguson Smith, as well as to my more immediate predecessors, my inspiring colleagues, Dr. Douglas Wilcox, Professor Mike Connor, and Dr. Maria Jackson, who are all now enjoying a very well-deserved retirement. I also owe a lot to my current and past colleagues in the NHS and the university, including Doctors Leah Martin and Saida Bhatti. I will now hand over to another current student on our MSc programme, Lavanya, who's going to chair the first session of today's fascinating programme of talks and discussions. Thank you.
Thank you, Professor Tobias. It was great to know about the history of our department. It is my pleasure to now introduce our first speaker, Dr. Amy Hausman from Cardiff University, whose research focuses on Timothy syndrome. Dr. Amy, we are looking forward to your talk.
Hi, everyone, and thank you to the Glasgow Genetic and Genomic Medicine for inviting Sophie and myself along today. I'll be giving you an overview of CACNA1C related disorders, which includes Timothy syndrome. So just a little background about myself. So my name is Dr. Amy Hausman. I have a first-class BSc genetics degree from a very small university called Edge Hill, up near Liverpool, or down if you're in Glasgow. So during my undergraduate degree, I went to Shanghai and I did a placement at Fudan University looking at the bioinformatic applications of genomics. I then completed my PhD in rare disease genomics at Cardiff University, and I finished that very recently, and have since started working for Timothy Syndrome Alliance as a research and public engagement officer. So I thought I'd introduce you to the gene CACNA1C and its associated disorders. So this slide shows you the big picture of how we move from a person with symptoms to an effective treatment. We start with a patient who, with certain symptoms, then we identify the genetic change, in this case, CACNA1C, and finding the variant gives us a name for the condition, but it doesn't always tell us the whole story. The biggest uncertainty is in the biology. We still need to understand what each variant actually does to the protein and how that leads to the symptoms that we see. Only when we understand that biology can we develop targeted therapies instead of just managing symptoms. So this pathway from patient to biology, patient to gene to biology to therapy shows both our progress and where the major gaps remain, understanding the biology well enough to design precise treatments. So what does CACNA1C do? Well, CACNA1C encodes the Cav1.2 L-type voltage-gated calcium channel, which is a bit of a mouthful. So what you can see here is the calcium channel embedded in the cell membrane. Its job is simple but incredibly important. It allows calcium ions to move from the outside of the cell into the cell, but only when the cell is voltage activated. Calcium carries a positive charge, you can see here with the 2+. So when the calcium flows into the cell, it changes the electrical state of the environment. That electrical change is what allows heart cells to beat in a coordinated way, neurons to fire, and many developmental processes to unfold properly. So this channel isn't just about moving calcium ions, it's controlling electrical signaling in some of the most excitable and sensitive tissues in the body. And we can detect how efficient a channel is being by using something called patch clamp analysis. So patch clamp allows us to measure the electrical current flowing through the channel when it opens. Because calcium ions carry a positive charge, we can detect those tiny electrical signals precisely. What this gives us is a direct readout of channel behavior. We can then ask, does the channel open more easily? Does it stay open too long? Does it allow too much or too little calcium into the cell? And these are really important for the function of the protein. So in patch clamp electrophysiology, we get a recording that looks something like this. So at minus 80 millivolts, the voltage gated calcium channel is closed. And when the channel detects a voltage stimulus, the channel opens and the calcium ions flood into the channel. And then we can see this as an increase in voltage. After a short time, the channel begins to close, and then bringing us back to a closed channel again at minus 80 millivolts. And this plot here also shows us how the calcium channel responds to changes in voltage. And importantly, that as the voltage increases outside of the channel, the likelihood of the channel opening also increases. So what happens in patients with a pathogenic variant in CACNA1C? Well, as you can see here, we've got the most common causative mutation for Timothy syndrome, and CACNA1C variants change how calcium channels open and close. So in CACNA1C related disorders like Timothy syndrome, we see that these processes are actually disrupted. So what happens here is that, for example, with this variant, is that you get a higher current for longer. It doesn't recover as fast. The voltage is a lot higher for longer. So therefore, it could be that the channel is staying open longer than it should be. So more calcium ions are entering the cell, which isn't always ideal. And we sometimes refer to mechanisms like this as the just right mechanism. Too much calcium isn't ideal, but neither is too little. We want it just right so that regulation of the calcium channel is really important. And what we see in CACNA1C related disorders is usually that these regulations are not as tightly controlled as they should be. And again, we can see here that even the threshold of the voltage is affected because of these CACNA1C variants. So why are these calcium ions important for tissues? Well, that calcium signal can cause different things depending on the different cell types. For example, they can make muscles contract, they can excite neurons, they can regulate gene expression, alter hormone or neurotransmitter release, and many more. So in any tissue that uses a voltage-gated calcium channel, calcium entry is the key step that links electrical signal to the cell's response. So where exactly is CACNA1C expressed? Well, this is a little plot that shows the expression levels of CACNA1C across numerous different tissues. As you can see here, there actually are quite a lot of peaks. So CACNA1C is expressed, for example, it's expressed in the lymph nodes, in the heart, in the skeletal muscle, in the small intestine. It's quite widespread, widely expressed in the body. So how might this translate to the symptoms that patients with CACNA1C variants have? Well, it means that patients with CACNA1C variants have a multi-system phenotype. And as you can see from this diagram here, we see that patients have neurological problems. So they often have epilepsy or seizures. They usually have neurodevelopmental disorders such as autism or ADHD or developmental delay. Structural cardiac abnormalities, prolonged QT intervals, so the recovery of the heart isn't working properly. They've also got cardiac arrhythmia, hyperglycemia, immune dysregulation, and even things like structural abnormalities like syndactyly, so the webbing of the fingers or the toes or the hands, and hip dysplasia. So really, and these are just the symptoms that we know of. So it's really broad because of the way that it's expressed. And this is a flow diagram that I personally think is really important. So it basically helps us to identify and diagnose CACNA1C related disorders a lot easier. So the key telltale signs in someone's symptoms that they may have a CACNA1C related disorder is that they have a prolonged QT interval and a neurodevelopmental disorder, such as developmental delay or autism. But as you can see here, patients present with many more symptoms spanning cardiac problems like bradycardia, structural cardiac malformations, and some of the things that I mentioned previously. With such a broad list of symptoms, it's really important that patients are monitored throughout their life, as some symptoms may develop later on. But what I really want to hammer home is, if a patient comes into your clinic with a prolonged QT interval and a neurodevelopmental disorder, please think, could this be CACNA1C related disorders? So I thought I'd just finish here by saying thank you for listening. And we recently have published a preprint about how to diagnose CACNA1C related disorders, including Timothy syndrome. So this could be quite instrumental for future patients and their guidelines for management. So yeah. Thank you. I'm happy to take questions now, or we can wait till after Sophie's talk because they might overlap with the question. I'll stop sharing.
Thank you, Amy, for that insightful talk. We will now take a few questions from the audience. You can post it in the chat box.
So I think we've got a question, which is, are there other calcium channels that can help compensate the CACNA1C loss? So the mechanism of CACNA1C related disorders, it's not that we know of right now, it's not really kind of a loss of function mechanism. It could be, but that's not been investigated at the moment. What we know is, at the moment, most variants that cause CACNA1C related disorders are missense disorders. So they're not necessarily removing the function, they're kind of a change of function. They're causing it to not quite work properly. And there's a lot of other genes related to genes involved in the calcium channels, like this CACNA1A, B, C, D, E, all of these, which can also be playing a role as well. But for now, it's not really the loss of CACNA1C, it's more, it's not working properly. And in differential diagnosis, what should be considered and what other disease is similar to this disorder? I think the problem with, well, not the problem, but the hard thing about a CACNA1C related disorder is that its symptoms are quite broad. So it could be that a patient comes into the clinic with a neurodevelopmental disorder and a CACNA1C variant, but they don't have they don't have cardiac symptoms. So sometimes we say that they've got a long QT type 8, which is also similar. So it's kind of quite hard to diagnose unless you haven't got that genetic variant. But yeah. And let's see.
Well, there is a question now coming to this CACNA1C gene-related disorders. Now, since it's related to various phenotypes and different disorders, does certain region, so let's say at the beginning of a gene, related to a certain phenotype, while another region related to another specific phenotype, or this is not the case here?
That's a really, really good question, and it's something that we're kind of trying to look at the moment. Currently, with Timothy syndrome, there's only a handful of really specific mutations that we know of that are definitely Timothy syndrome. There's so many more CACNA1C variants that don't necessarily have the same phenotypes as those specific Timothy syndrome variants. But when it comes to kind of the regions, at the moment, what we're hoping to do is kind of map all of the variants, the mutations onto the gene, but it's quite hard because there's loads of different isoforms of CACNA1C. So sometimes different areas of the brain will express different transcripts. So it can be quite tricky. But at the moment, there are some specific Timothy syndrome like hotspots, but the rest of it's kind of unknown. And that's kind of what we as a Timothy Syndrome Alliance are trying to figure out. But yeah, there's a lot of unknowns, but that's definitely something that we're looking at. And I probably would say there probably are some phenotype specific regions, but we just haven't looked into them enough yet.
Thank you.
Hi, so there's another question that asks, which population is it commonly affected?
Yeah, so it's kind of, we don't really have kind of ethnicity specific or population specific variants at the moment, because we're kind of finding patients with CACNA1C variants kind of all around the world at the moment. So we've got we've got loads of Brazilian patients, we've got patients from Spain, Italy, USA, UK. They're kind of all over, but in really small numbers. So at the moment, it's not really kind of, it's because it's autosomal dominant or de novo, it's not kind of as easy to track as, say, like a recessive disorder where it's more prevalent in certain populations at the moment. Yeah.
And thank you. There's also another question that asks, is it curable, or is there any treatments method that is currently going on?
Yeah, so at the moment, so that the main goal would to be get some. At the moment, no, it's not curable, and it's we're working, hopefully, in a couple of 5-10 years that there will be a treatment available. But because the biology is kind of quite unknown at the moment, treatment is kind of symptom specific. So it'll be kind of treating the cardiac problems with like a pacemaker or things like that, or like treating the hypoglycemia with like insulin. It's not necessarily kind of a systemic gene therapy at the moment, but hopefully in the future, maybe, fingers crossed, there would be something that could be more beneficial to the patients.
Hopefully. I think the last question for you today is, what made you interested in studying about this disorder?
Yeah, so for me, I think, well, I came into Timothy Syndrome Alliance from a rare disease background anyway. My PhD wasn't really in neurodevelopmental disorders, so it was more in rare colorectal polyposis syndromes. But for me, it's kind of just the rare disease aspect. I feel like what's the point in doing the research if it isn't benefiting the patients? So when I saw the opportunity with Timothy Syndrome Alliance, that's exactly what they do at the charity, helping with the trying to benefit patients as much as possible who've got this really rare disease. So for me, it's kind of my morals and my interests aligned just to helping rare disease patients, but especially now that I can see how important it is for CACNA1C patients.
Thank you for answering the questions, Amy. We will forward you if there are any other questions. Moving on to our next speaker, Ms. Sophie Muir, the CEO of Timothy Syndrome Alliance. Thank you for joining us, Sophie. We are looking forward to your talk.
Thank you very much. Okay, so thank you for having me and for the opportunity to share my lived experience. Calvin is my middle son, sandwiched between my other two boys. When he was younger, he was a little bundle of curly ginger hair with the cutest Harry Potter wire glasses and two fingers always in his mouth, seeking out comfort. He'd get my attention with these sounds that would make you look at him so he could communicate with his facial expressions, along with pointing. Of course, I knew what he was trying to communicate most of the time. You see, we both used a form of sign language called Makaton that's done alongside speech to help promote its use. I'd started learning foundation level when he was 3 and quickly moved to enhanced because he didn't speak at all. Nothing, just I didn't know if he was hungry or if he was thirsty. I didn't know if he wanted to line up his cars or cuddle and look at his pictures in a book. I didn't understand what he wanted. This was leading to outbursts from him and tears from me. Meanwhile, we're repeatedly being told that he'd soon catch up and we were sure that everything would be fine. I taught Calvin the signs and the rest of the family in the nursery school in an attempt to make him fit in and for others to communicate with him. Even his support workers at school were Makaton trained because it turned out Calvin really didn't start speaking words until he was around 7, by which time he called needles pins as he'd had so many stuck in him for so many tests. Chromosome, Angelman syndrome, Fragile X, all negative. Of course, I'd done my fair share of late night info Googling his symptoms too, trying to join the dots. You just know that something isn't right. It was the not knowing that was the hardest bit. No one can pigeonhole it. We didn't know what to do to make Calvin's life as easy or productive for him as possible. It had become more obvious as Calvin was growing that he had a wide range of challenges. Even though the additional support Calvin had been receiving at school was full-time, the very low range of scores in all his assessments showed that Calvin was floundering and couldn't access the curriculum. It was clear that he had significant needs in all cognitive areas and severely delayed and disordered development of expressive and receptive language. His extremely slow progress was not equivalent to his chronological age. The gap between Calvin and his peers was growing. Most notable was that Calvin was being overtaken by his brother, who is 4 years younger than him. In 2016, when I received the genetic results by post, confirming a CACNA1C variant, I'd just spent the last 8 months battling with the Education Authority again. So here's the timeline. At the age of 6, we joined DDD study, which is deciphering developmental delay. His symptoms were hypertonia, abnormality of movement, delayed speech and language development, and drooling. When we got the results just before he was 10, his symptoms were hypertonia, complex tics, delayed speech, verbal dyspraxia or apraxia of speech as they call it now, expressive language delay, speech disfluency, that's stuttering, ADHD, anxiety, neurodevelopmental delay, coordination disorder, autistic behavior, fluctuating temperature, he goes down to 35 degrees for no reason, prolonged QTC, abnormal T-wave, right bundle branch block, and astigmatism. Very quickly, I learned that his CACNA1C finding was a VUS. Variants of uncertain significance are genetic changes with unclear health impacts, meaning it's uncertain whether they contribute to disease or are harmless variations. It's only because we were in a research study that I was told the result. VUS findings are not reported through NHS testing. My biggest challenge was that VUS findings cannot be used for clinical management or treatment decisions. So here began my biggest, steepest learning curve. In 2017, I joined the Imagine ID study. That is a study researching intellectual disability with genetic cause. 2018, I'd found a small group of families with Timothy syndrome. At that time, Timothy syndrome was the G406R variant, and that was presenting with autism, syndactyly or hip dysplasia, prolonged QT, and early onset arrhythmia. But he wasn't that variant, so he had no name. In 2019, we were called to give bloods by the Imagine ID study. We went to Cardiff, where I soon realised they'd been studying both common and rare variants of CATNA1C for the last 10 years. I decided to register a charity to find other families just like us so we could gain knowledge and attract attention from industry, pharma, bio. That December, we had a family day where I brought together 8 families with no budget. And we've been working closely with Cardiff ever since. So at that time in 2019, there were 43 known global living individuals with CATNA1C globally. Calvin was what they call an N equals 1, so there was only one known of him globally. And we got the results back from the 100,000 Genome Project confirming CATNA1C. In 2020, we made a film called Timothy Syndrome Alliance to try and raise awareness, and I entered that into some festivals and we won awards with that film. And then COVID hit. Everything went online. In hindsight, that was probably beneficial because it enabled me to meet people online. Built a website with a lovely volunteer who's actually still with us, had a conference online, and made another film called Rare Strikes Back, which was born of frustration from rare disease research going onto ice while COVID was taking up everyone's attention. By 2021, we had an online phenotype-genotype research collaboration with Cardiff and Stanford, and we had another conference, Brain Research Conference. By that November, I was confident enough with the evidence that I'd found to request reclassification of Calvin's variant. This was based on iPSC cells showing mixed function. They were the ones that we donated bloods to Cardiff. I'd identified an individual with a pathogenic variant in the same amino acid as my son, which had not been found previously, as there'd been changes since the paper was published in how the genome was annotated. I had confirmation his variant was located in the IRES-like region of the gene. It's also located within 68 nucleotides that are 100% conserved between human, rat, and mouse, suggesting its importance. And I'd also found his N equals 2. I was in contact with the clinician of an individual with a likely pathogenic variant exactly the same as his. In 2022, we launched the CATNA1C Community Registry. That allows access globally. We started exploring the psychosocial impact of CATNA1C on families. We began speed dating industry, and we won our first PPIE award. I also co-founded the Voltage Gated Calcium Channel Collective, which is a collaboration of all 10 genes representing the calcium channelopathies. So a year later, in that December, at age 15, I finally got confirmation that Calvin had been reclassified as likely pathogenic meaning disease-causing. 2023, we successfully started adding panels to Panel App. We added epilepsy and hypoglycemia. We launched our scientific advisory boards, and that is chaired by Dr. Jack Underwood, who is a clinical lecturer in forensic psychiatry. The paper from the study that we'd done, set up during COVID, was published, Levy et al. We had a language accessible conference. We started working with Helix on a Wikipedia CATNA1C related disorder page. And we set up our Rare Minds counselling service that we called Mind the Gap for our families, individuals and couples. 2024, we had a film called Connections that we'd made with Cardiff University that came out that reveals the vital network between families affected by Cat 1C and the researchers striving to understand them. 2024 was also a significant year because we were recipients of a grant from Chan Zuckerberg Initiative, Rare as One Network. We were cycle 3. That was $800,000 over 5 years. That is specifically for us to build capacity, convene our community, and strengthen our research network. 2025, we won another award. We got highly commended with Rare Disease Research UK, and Jack won Best Public Engagement at the Cardiff University School of Medicine. We had another online conference, 70 plus attendees from 19 countries. Again, that was multi-language. We've built our mission support officer by building capacity. We're able to do newsletters now. We've got the consensus out under review with the European Journal of Human Genetics. As Amy said, that's nailing down a clear, solid definition of Timothy syndrome while introducing a more inclusive term for those that have different conditions that don't necessarily present with cardiac. We are building a portal to provide a unified resource for characterising the clinical spectrum of CRDs, linking specific genetic variants with their phenotypic manifestations, along with output from variant modelling. So that will support clinical variant interpretation, enhance patient care management, and identify biomarkers for clinical trials. We grew an Amy. We're delighted that Amy's on the team. And we're 2026 now. So we've just won another PPIE award, the Best Research Partnership Award. The registry, we're doing a data pool so we can analyse and publish the data. That's currently got 123 participants. We're having an in-person conference. We've got families signed up now from Italy, Germany, USA, UK, Australia, Spain, Brazil, Norway, and the Netherlands. We're setting up a biobank, and we've also raised enough funds to start our speech and language global phenotyping research study. That's with the MCRI in Australia. So I am now part of and advocating on behalf of a global community of more than 250 families. So we've grown a lot. We have a growing and wonderful network of researchers and clinicians, and lots of plans underway to keep ethical, scalable progress in our rare disease. Calvin is now 18 and attends an amazing SEN college. And I'm still having to learn new things like how to navigate mental health capacity assessments. I hope that gives you a kind of overview as to what is going on. And I'm happy to take questions.
Thank you for sharing your experience with us, Sophie. Your strength and dedication is truly admirable. We will now take questions.
Thank you so much. There's one question. So this person said you had to wait many years for a diagnosis. How much has the speed of diagnosis improved for children who are just starting to show signs?
It depends it depends how they're presenting, to start off, as to whether it gets picked up. It depends on the knowledge of the clinician and the healthcare teams. It depends what they find when they do genetic testing. If it's a VUS, you can end up in a world revolving like me. It's kind of no man's land, particularly if it's a variant that's not been seen before, which is going to happen because we're a rare disease. So it's really important that these findings are uploaded to ClinVar, that labs submit from all over the world and help empower the research that we're all doing together along with Cardiff and the other collaborations that we've got with Oxford University, Earlham, Stanford. I mean, it really is all coming together to find the answers together. It's a teamwork job, isn't it?
Thank you so much. I don't think we have any more questions for now. If we do, we'll send you an email. Thank you.
Okay, everyone is thanking you for sharing your great journey and putting in a lot of efforts in the research as well to you and your team. Okay, so in the interest of time, thank you for answering the questions. Passing on to Bettina to introduce our next speaker.
Thank you, Lavanya, and thank you, Dr. Amy Hasman and Sophie Meyer, for your wonderful talks. Our next speaker is Dr. Haya Al-Balul. She's a senior specialist in molecular genetics and genomics at the Kuwait Medical Genetics Center. We're very pleased to have her with us and look forward to hearing her talk on cystic fibrosis.
that you got a short, refreshing break. And thank you all for joining us on this fireside chat based on genomic equity and global clinical perspectives. As we all know, genetics is advancing rapidly across the world, but the way it is practiced, taught, and integrated into healthcare systems can look very different depending on geography, resources, and local needs. Today's session is an opportunity for us to hear directly from specialists working in different parts of the world and to understand what clinical genetics looks like in real life practice. I am Dr. Shweta Matthews.
I'm Susanna Coronel.
And we are master students in medical genetics and genomics. And today we are the moderators of the session. Today, I would like to welcome our excellent panel, Dr. Ahmed Al-Akil, a consultant clinical geneticist working in Kuwait OL Hospital. We are also joined by Dr. Sasane Patritifan, a physician with dual training in hematology and medical genetics from Thailand, and Dr. Catherine Mutinda, a consultant pediatric geneticist working in Kenya with a focus on genomic medicine and equitable access to care. Through their experiences, today we hope to explore their career journeys, the realities of practicing genetics in different healthcare systems, and the challenges and opportunities in advancing genomic medicine globally. To start off with, we'll be having a few base questions. And once our speakers are able to communicate the answers for those to all of us, we'll be opening up the fireside chat for receiving questions from our beloved audience. And thank you again to the speakers for being here, taking their time off super busy schedules, I'm sure, and for sharing their experiences with us. So let's get started.
Okay, first of all, we would like our speakers to introduce themselves. So please, Dr. Ahmed Al-Akil.
So hello everybody, my name is Ahmed Al-Akil. I'm a clinical geneticist from Kuwait. So my background is I graduated from Kuwait University Faculty of Medicine back in 2002. I don't know how old you were, but it was 2002. Then I got my training in Germany from 2007 to 2013 in Münster University Hospital, Institute of Human Genetics, where I received my German Medical Board of in Human Genetics. I also have a postgraduate diploma from Manchester University in clinical bioinformatics. I'm responsible for genetic services at Kuwait Oil Company Hospital. So that's what I'm doing right now. My interest is dysmorphology and bioinformatics. Thank you.
Okay, thank you so much. Please, Dr. Sasini Patritifan.
Hello, I am a based on hematologist, and after finished training in hematologist, I'm also interested in medical genetics, especially genome therapeutic or something like that. So I start study the certificate in medical genetics before came to the Glasgow. Yeah, and I just graduated from University of Glasgow last year. Now I am working as an hematologist and medical geneticist at National Cancer Institute, which under Division of Medical Service. We responsibility and take after about some policy, yes, of the medical genetic that are launching in Thailand now. Yes, so I saw something like inequality between the full service and the full facility under medical school and the real practice in the rural area or in the central shim of Thailand.
Very interesting. Thank you so much, Dr. Sasini. Now, please, Dr. Catherine Mutinda.
Good afternoon, everybody. I am so glad to be here. I was in your place in 2020 during the pandemic, and I'm an alumni of your school and an alumni of this particular training. I studied my medicine in Charles University, Prague. I graduated in 2006. I hope that doesn't give away our ages. Then I did M.Med. Pediatrics here in Nairobi at the University of Nairobi, graduated in 2013, and then moved on into genetics in 2020. That's on on the part of learning. On practice, I'm both in public and private practice. I enjoy so much dealing with babies, and I think that's why I started into pediatrics, and I also went into rare diseases, which are generally, as we had the lived stories around children. So I wanted to offer answers, and I think and I believe I am offering answers. Glad to be here. Thank you, Ed. Thank you, Saida. Thank you, Maria Jackson. I'm forever grateful for this. Thank you. Over to you.
Thank you all for letting us know about your personal journeys. And next question is, how does the healthcare system, especially regarding genetics, in your respective countries operate? I think we can start off in the previous order as well. Dr. Alakil.
Thank you. So usually the case come from either pediatrics or reproductive genetics, which is IVF mainly, obs-gynae, and there are some cancer, the malignancy cases. So through this, the genetic counseling, construction, taking the history, constructing a pedigree, and after that, put the roadmap of the genetic or genomic testing, and from there we go to the when the result come, depending on the result, the management will be construct. It's as simple as it is. And then when the management, the treatment is started, the management also in combination with the multidisciplinary medical team. So this is how basically the things going in our cases.
Thank you so much. Dr. Safanipa, please.
For the introduction, the system operate in Thailand. I think it's like NHS in UK, but it's not the same. We have something like universal health coverage, which cover about 80% of the people in Thailand. Another is likely civil servant and social security scheme with a few percent, but all of them cannot access to the genetic service. Maybe except the BRCA in breast cancer, which just launched a few years ago, and newborn screening only in the metabolic part that not genetic like plasma amino acids only. So all patients that have to access genetic testing have a choice only in large medical school in central area of Thailand, or maybe under the research Thai, yes, to save their money, or maybe another choice go to the private practice. So this is maybe one of the problem, but now there are many of team that develop to compensate and develop of this thing. So I am practice both in National Cancer Institute under government section and in the private practice. So I saw that this of inequality that is a problem of the to access the genetic testing of Thailand. And another problem that I think we still have in our country is a very new thing. There are no official training of the geneticists in Thailand, maybe except of in the pediatric department, but not in the medicine department. So everyone have to study abroad. And the knowledge of the clinical genetic in the undergraduate is still limited in overall.
Thank you so much for letting us know about the situation, how it is in Thailand. And now we have Dr. Catherine Mutinda. Thank you so much.
Thank you, Maria. In Kenya, just like in Thailand, I think, there is no official framework on how genomics and genetics is practiced. That's my life mission, to make it practiced here at home. And how am I doing this? I am teaching in every opportunity I get. I am shouting out and talking. So as my colleague in Thailand has said, that it happens a lot in pediatrics. We have managed to penetrate now into medicine. I teach also the master students in medicine. I have also started teaching the undergraduates and we have just started a fetal maternal fellowship, and I'm right there. So education is one of the ways that we are getting there. The second thing I wanted to talk about is how the clients are accessing care. So two ways they're accessing care. One, privately, to my clinic. I run a genetics clinic on Tuesdays and on Thursdays. I am very overwhelmed with the work, and therefore I have chosen to also apprentice two other people who had done genetics as a bachelor's and sent them to my colleagues in India to have them train around genetic counseling. So I have one already fully baked back in the country and one out there getting some experience so that I can have sort of a team that starts, yes, in the private sector, but we do have some free sessions for some patients in the public sector. I do work in the public sector, so I do consult for them as well. Then there's the research angle. There's a lot of research happening. I am glad to have heard from my colleague in Kuwait about cystic fibrosis. We are opening up a database for cystic fibrosis in Africa. It's opening up also a database on retinoblastoma genetics. We are talking about genetics of so many things like inborn errors of metabolism. So it's everywhere and trying to get a lot of support from everywhere that we can, a lot of people to work with us. So the final aspect is laboratory support. As you know, there are no set up molecular laboratories in Kenya and in Africa at large, except somewhere in Egypt and a bit in South Africa. So we heavily depend, or when I came back, I heavily depended on America, on Invitae, and then it was absorbed into LabCorp, making it a little bit difficult. So we have moved into the Indian market. They've been accommodating to us, and we are working heavily with Newberg and New Genome in India. So hopefully we had a conversation just this weekend with Newberg, and we have our variants now being deposited in OMIM, what we are finding, and hopefully now we can have some diagnostic daisies coming down in the rare diseases forum. Thank you, and over to you.
Thank you so much, Dr. Muthinder. It makes us feel so proud to see an old alumni of our program going out into the world and becoming such a strong advocate back in their own home countries. Thank you so much for answering the question.
Now, our next question is, how are genomic services supported in your countries? First, Dr. Amar Alki.
So in Kuwait, for the citizens, it is covered. So it's as simple. Whether they are, so the government covers all genetic testing. This including from karyotype to fish, to single genes, to familial pathogenic variants, to NGS, which is whole exome, whole genome, and chromosome microarray, the genomic testing. Now, for the non-Kuwaiti, it depends. If they work in the oil sector as I work there, it is also free of charge, whether they are citizen or non-citizen, since they are employed. So in the oil sector, so KOC Hospital or Kuwait Oil Company Hospital will cover it. Now, for non-Kuwaiti, government has taken over and they pay, just a slight around 10 to 12% of the cost. So that's basically how things coming. Almost all tests are covered.
Well, that's amazing to know. It's great. It's fabulous. Okay, well, same question for Dr. Sasinita.
Yes. My talk in this question maybe look at the total stage of country, but just to share what we do in the initial stage. Yeah. If patient have some genetic disease, of course there are no much of choice that they can do except go on the large academic hospital and wait for some research time or something that can support their diagnosis. Yeah, and the other is go to the private hospital, but private hospital in Thailand is affordable. Yes, it's maybe not so expensive when compared with some country. Yeah, but in the real practice of the early stage, we start with the pharmacogenomic and the cancer genomic, like the breast cancer, BRCA, and cancer panel under all of the government coverage. Yeah, but and we also have the Genomic Thailand that launched around I think it's 5 years ago and just finished and in the process of the publishing. Yeah, but we found that the problem in this initial stage is the lack of human resource like the bioinformatic, genetic counselor, yes, or some physician that know about this. So, although we have a genomic data like BRCA or TP53 mutation, but we don't have a genetic counselor that can push this information to the patient. Yeah, so we start training. It's like a short course training of the genetic counselor. But the problem that follow it is, I think, genetic is something rare disease. Yeah, so it still need an experience in under the correct supervision. So what I do and we do now under my practice is we open for the tele consultation in the some rare case. It's not so rare, but it's like uncommon cancer variants, something like that. So if someone in the rural area hospital have a question about this, they can submit their case and we will open the conference around 2 or 1 time per month, yes, to answer about this. Yeah, and this is still in the developing town. Yeah.
I guess it's me, right?
Yes. Thank you.
So Kenya, like England, had the National Health Insurance Fund that has now moved to something we call the National Social Health Authority. So that morph has meant a lot of gains have been lost, unfortunately. And we are starting all over again to start advocating for some things to be covered. Originally, the NHIF would cover cancer genomics, and it is part of the policy. Rare disorders, unfortunately, are not covered. They have no financial support. A lot of Kenyans do have private health coverage, like insurance covers, which also consider rare disorders congenital, therefore not covered. So most of the clients we see are paying out of pocket. And it's since 2020 when I came back, the prices are going down and we hope to push them even lower. We have a lot of collaborations. I forgot to say I also sit in the ethics committee. So I end up meeting a lot of people who can collaborate and help some people who are in need and help grow the science further. So that's how it's supported. We do have very, very huge community of bioinformaticians. As I said earlier, we are growing the genetic counseling group. We are hoping to have a 360 as we move ahead. And yeah, looking for friends out there. Thank you.
Thank you so much.
All right, thank you, everyone. Again, the next question that we have is, what are some of the social or cultural factors that determine whether a person accesses genomic healthcare in your particular country? We'll start with Dr. Alakir.
Well, fortunately, with increase the education level in the Arabian Peninsula and Arabian Gulf, people really accept more and more, to go to genetic clinic and checking for their children. Not only that, they are going even for the screening part, for the preventive part, this is really, really good. However, even with this positive, we still have a high consanguinity rate. And this will increase the autosomal recessive with rare disorders. So we still have this, for example, in Kuwait, we still have 50% consanguinity rate. We still, you know. I think the Saudi Arabia also have, it's going down. Saudi Arabia, Qatar also have also around 50%, 51%, which is similar to Kuwait. Bahrain is even more, it's 60%. However, they are willing now to go more than before, and before they were really don't want to go there because they don't want to get this family stigmatized with a disorder that, if we married from them, maybe we will have also the genetic disorder coming to our side on. So that was before, I'm talking about, 2004 to the, and even when I was a student, when you were so young, that back in the 90s. Now it's different. Now they are really educated. Okay, we know there is a it will not genetic disease. It will not automatically transfer to another family if we married this or that. So they are willing to go to the genetic side for genetic counseling and prevention. Now, this also apply, to use the prevention before marriage and even after marriage, for the prenatal diagnosis and a pre-implantation testing. They are really now open. And actually, if you see my clinic, I have 3 clinics a week. One of them is the reproductive genetic clinic for the pre-genetic diagnosis and pre-genetic testing. So this is how people now shift because of the level of education, which is really good. And if you prevent is far better than having treating a patient. If you prevent, for example, spinal muscular atrophy, this one example, the gene therapy is available. It's far better than having an affected child and give him a new gene therapy. Maybe he's cured, but we don't know about the long term now. So prevention is far better than cure. Thank you.
Thank you so much, Dr. Alakil. I think we'll move on to Dr. Sasanipa next.
Yeah. We have something like Dr. Ahmed. Yeah, but in my opinion, I have something that is a different point. We have both the social determinant that increase and decrease access to the genomic healthcare. Yeah, we will start from the something that maybe decrease or under access to the genomic. Yeah, the first thing is geography and socioeconomic status, because the genetic service is most located in the Bangkok or in the center of East, Northern and Southern. So people have to have require out of pocket money for the genetic testing and also for the travel costs and accommodation to go to the Bangkok, that is the center of Thailand. Yes, another is maybe educational and health literacy. Yeah, because maybe some people still not understand well about genetic disease or not realize the importance of testing that maybe prevent the sequelae or precise management of this condition. But in this era that the social media arise, yeah, we think that we have a social media to influence the patient to access to the genetic testing. Yes, like when someone like matter type, we have a children with the Turner syndrome. Yeah, so this one is arise some important or something like interest of what it is, and is do we should abortion of this? Yeah, so this is that something like opportunity that we can give an education to the people under that circumstance. Yeah, and another thing that we saw you talk about the SMA. We also have an situation that in the it's a carrier screening in the pre-gestational, because the carrier screening is not still standard in the Thailand practical because it's not under the government practice. So it's not all of case that we can do a carrier screening. And some people have a child, and their child was born with the SMA. And after that they found that their child should treat by the gene therapy or something like that. And the father arises with the question, why the carrier screening not be all of the patient or all of the population in this country? And I think this is a small voice to raise the importance or raise a recognition to anyone that have an authority. So now I think we are in the state that social determinants such as the recognition from social media, increased awareness, and also the Rare Disease Day that also have in Thailand.
Thank you, Dr. Sasanipa, about that. I think the added advantage of social media is also about spreading these awareness of rare diseases.
Oh, and can I speak one more?
Yeah, sure.
It's about maybe we can exchange an experience about the genetic discrimination act like Gina. Because Thailand not have this act, so many people like deny to have a genetic testing because aware of deny from the insurance. We still don't know how to manage with this.
Thank you so much again, Dr. Sasanipa. We'll be moving on to Dr. Catherine Mutanda now.
Yeah, thank you very much. So we in Africa are known for our traditional medicine, our traditional ways, our religious ways. So stigma is real here. Unlike Ahmed's side, Africans are not consanguineous, but we have consanguineous group from our Somali counterparts who are part of our population, which is very different. So we do have rare disorders. They are considered by most cultures as a curse, and things are getting better. How are they getting better? As my colleague from Thailand has talked about social media, I have a podcast called Code of Life, and interestingly, I took care of someone who is in the same realm as Victoria Beckham here at home. She's a musician, and she came for an NIPT. She was pregnant in her almost 50 years old, and she came for an NIPT, and she pushed me to start the podcast Code of Life. And we've had so many patients come and discuss their rare disorders, the SMAs. We do have a very large group of SMA patients in Kenya. Sickle cell disease, discussing a lot. It's a long podcast, and we're still creating more of the series. So that has helped patients actually start doing genetic testing and also coming into the clinic. Unfortunately, the downside has come to be direct consumer testing. We have people importing kits, sending them out to Ancestry and the rest of the databases who are doing direct consumer testing. And we've had to write, I have had to write pieces of information that Ancestry is not necessarily giving you a diagnostic. So there is that overexpression of interest into doing genetic testing, lifestyle, but we do not have enough database background for the African group to have a good outcome so that we can actually maybe benefit from the same. So a bit of work going there. So those are coming up. The other thing is our African traditions, as much as they were non-literal marriage, one of the biggest things, and I'm preparing for one tomorrow, is to go for what we call a pre-marriage ceremony, where the families meet and draw a family pentagram. So that was a tradition in Africa. So you say who you are, who your parents are, how they're related, and there's this whole group of people who are discussing whether that marriage is acceptable. And usually they're looking for disorders, diseases. So with that in mind, that being a culture in most of the African traditions, it's very easy to sell genetic testing. It's very easy to sell genomics. And that's why the access is becoming better and very easily acceptable. I think I've covered most of it. Yeah, we have very big advocacy group, RDK Kenya. I hope you all joined me on Wednesday. I was celebrating with RDK Kenya, the Rare Diseases Day. And yeah, things are getting better. Thank you.
Thank you so much to everyone on the panel. We'll be moving on to the next question.
So our next question is, what do you see as the most urgent priority for genomic medicine development in your country over the next 5 to 10 years? We start with Dr. Alakil.
I think now at this time, let's see now where we are now, and from here we can see the future. So the good news is we have two things. So one is between Arabian Gulf, especially what is called the GCC countries, the Gulf Countries Council countries, which is Kuwait, Saudi Arabia, Bahrain, Qatar, United Arab Emirates, and Oman. So 6 countries. So we already have, a WhatsApp group coming together, on daily basis. We talk to each other, on daily basis, sometimes by voice messages and everything. And this, although it seems very small, it has a tremendous impact. Tremendous impact. I will give you an example for you and for everyone. In 2022, I have a patient. So when I saw him, I didn't I didn't construct any, suspected diagnosis or a short list of differential diagnosis to make, for example, a panel or something. So I start, like the usual chromosomal microarray. And it was negative. Then I did the whole exome because I search, in the literature, still I couldn't find, a short list differential diagnosis. Then after that, it came a pathogenic variant and everything's. Now, after, because of rare, because we have high consanguinity, as I said a few minutes ago, extreme rare syndromes do exist in our region. So when I talk to them, I shared this, that I have a patient that has this, and we found this pathogenic variant. A colleague of mine in Saudi Arabia said, yeah, we have this. So we talk privately, through Zoom, just like now. Or it was the same tribe. So now when I have someone, one of the things that, okay, I have this and this. Did you see this similar case? And the same thing with me. This is tremendous because now usually I do 65%, around 2/3 with microarray whole exome, and then I'm shifting now to whole genome, directly to whole genome. Now I have 50%. So the 15% out of 65 was solved just by getting with each other, because we have similar tribes, similar families, similar genetic pool. Now, I think for the future, this should be even increase more, to include, Africa, international with you guys and everyone, this will give a tremendous power for everyone. Now, I think also the second thing is what we lack now. I think in the near future now, I think unfortunately till now we don't have a Kuwaiti genome database. We don't. We have a draft. I think this should come, and we should, have a joint venture with everyone. Because in Saudi Arabia, in the Middle East, Saudi Arabia, they do have their database, Qatar, United Arab Emirates, and soon Oman, very soon. So I think in the future now, we can help our neighbors, for example, at least in the Eastern Africa, Northern Africa, to help each other, to get each country's database. This in turn will help everyone, to have a better understanding, intimacy syndrome, our great doctor, who showed us, how the gene and how we get this and the whole exome and the whole genome. I think, the better is we have the getting together, before even start, because now genomic testing is not difficult to do. For example, in 2025, the decision of our hospital to shift to whole genome, it wasn't difficult because the cost become less, the accuracy become even better. So I think for the future, I think for every clinical, for every human genetic service is to collide, and I think Corona taught us very well the power of online meeting.
Well, thank you so much. I completely agree that that's the future. Like, we should help each other as colleagues. Definitely, I completely agree with you, Dr. Alakil. So now, please, Dr. Sasinipa.
Yes, we have religious and cultural. For the religious, mainly Thailand and maybe in the Southeast Asia around Thailand, the main religious is the Buddhist, with believe in something like karma. Yes, so some people still believe any abnormality that was born with you is something like karma and irreversible. And yes, and it's something like stigma that you cannot change it. But now the education and the social media or online awareness event, yes, can raise this understand that it's not a karma. So I think we try to promote that it's just some variable. Yes. For the one of the interesting example that I do now is about the sex chromosome. Yeah. We believe that we have a case that some mother have a children with sex chromosome still think that do they should to terminate that baby. Yes, but we try to announce that sex chromosome abnormality is just a variable, and it's same as something like the tall stature and the short stature is only the variability in the height. Yeah, so we try to tell this is a normal one boy or normal one girl, that they maybe need some extra special care from the doctor. Yeah, and not anyone's fault. Yes, and for the cultural consideration, we think as I have some patient from Southeast Asia in my private practice that is an international clinic, we found that the inequality in the social race, the social classification is a social class segregation. So parents still need their child to be something like perfect. So if there are anything not perfect in the prenatal stage, the thing about the termination will raise in many cases. That so I think the first thing that have to be raise of awareness in our country maybe about is like through understanding about genetic disease, that is only some illness that can happen with anyone, and we can prevent after sequelae, can precise management and make the patient have a good quality of life. Yeah, that we try to do now.
Well, thank you so much, Dr. Sasinipa. Please, Dr. Catherine Mutunda.
So where do I see our future? I see a huge genetic service in my country. I see a situation like what Ahmed has described, a database. A database will help us stop talking about rare and start talking about uniqueness, and start talking about pharmacogenomics, start talking about drug discovery and so on and so forth. So I also hope and pray that we'll create teams like Ahmed has discussed across Africa, across regions, to discuss some things that are very rare. I recently diagnosed a baby with a trisomy 22 and found out that that was the 20th trisomy 22 that has been reported globally. But it would be so much better if we could all talk together, as Ahmed is talking with his colleagues in the other 6 countries. So as a genomics family, as what the late Professor Ferguson had dreamt of, create a unique family where we can actually discuss, describe, and also create a better future where we are more of preventative than reactive. And if we cannot prevent, then actually go into the treatment modalities, make them available to our clients. Thank you.
Thank you so much. I think it's completely true. Well, it's better to prevent than anything. So completely agree.
Thank you so much to our wonderful panel. And those were our prepared questions and the questions that we had already received prior to the fireside chat. Next, we'll be opening up for questions from anyone in our beloved audience who want to ask anything to our panel members. We do have one question now from Maria Jackson. Huda will be asking the questions to our panel. Giving over the stage to Huda. Thank you so much.
Thank you, guys. So for the first question that we have for our panels today is, how effectively do the available genetic services reach the rural populations? Can we start with Dr. Lakil?
So if you know Kuwait is smaller than London, now you realize we don't have a rural places. It's so small. It's so small. However, I ask my colleagues in Saudi Arabia, before it was a bit difficult, but now with courier and new roads and these kind of things, sending sample is not difficult in our area. However, in some area is difficult. For example, in Iraq, it's not easy, to send because of the roads and everything. They told me, but now it's better. When I talk to my colleague in Iraq, the same thing, in Oman, they told me that because of the nature of the mountains, so they reach, but still, how. Fortunately, with getting, that's the wonder of things, when you address the problem and then try to solve it um, in a flexible way. For example, in Oman, they have some difficulties, in sending, from rural places, especially in the mountains. What they did is, okay, we don't put a genetic service there because still, it's a rural. So what they did is they did what is a very good thing, DNA banking and DNA. So they send a DNA sample, not a blood sample, because DNA is more stable and much less, to pack them together, for different patient to send them to the city. So in Oman, this is a very excellent, an example from them and how flexible. So they extract the DNA in their major labs. This is what I heard, in the rural side. And many things solved, because of this. The second thing is, again, what Corona taught us, the power of online meeting and online, evaluation. Before we said, oh, it's impossible. Now you can have a full degree, a full master degree online in bioinformatics. Okay, so this is how powerful the online is. So this is also very helpful, because when I ask my colleagues in Egypt, for example, I asked a professor, in the Middle East, one of the famous professor in our region, Professor Najib, Kamal Najib, and he told me, I receive many online, consultation, you know, from the rural areas and in Egypt. And Egypt's a huge country with mountains, desert, south. No, you know this very well. So uh, I think with try to be flexible, in solving things. Things will be solved, in a much better and more accurate way. So in Kuwait, summary, we don't face this big problem because Kuwait is smaller than London. So we are so small. So what is rural side in our, you just take a car and go bus and you reach everywhere. In Saudi Arabia, it's the same because they have a very good, excellent places. Oman, they fix it, by sending. I will just give an example of what in our area. The same thing with Egypt. So that's basically what I believe. And hopefully, again, be flexible, in solving a problem. It will be solved in a very accurate and with high quality.
Would any of the other two panel members like to add on to that, please?
In Thailand, we have a private lab provider, yes, that can cooperate in the rural area hospital and the center. So the specialist in any of rural area hospital can send for this genetic testing under some consultation with the geneticists from the center. And after they receive a result, they can consult by the online. Yeah.
Thank you very much for the question. Maria was my teacher, so I hope I'm doing you good. For the rural population, even for the urban population in Nairobi, it's not that easy. We start with the financial impediment. I am the only one practicing and practice twice a week. Of course, you know the burden of carrying so many stories in your head. And also social issues. There are legal issues that's around the same. I'm also battling with ethical issues around what are we going to do about it? So you tell me my child has Porteous syndrome. That was very exciting for the mother because she finally had an answer. But now what next? Now I have to talk to all my colleagues and educate them and then start that process of treatment. So I have tried to be as flexible as I have met and tried to do some online consultation, which I shortly after I started stopped because there wasn't support for the patient on the other side of the screen. So I gone back to the drawing board and I'd like to create more teams so that they are so far away. I did an online counseling session for a patient who was in Somaliland, and as we were talking, and it was in such a good situation, as we were talking, screen went blank, she went off, and there was nobody to tell us what's going on the other side. It's never left me, and I've not yet done another online consultation since then. So I will work on trying to get Ahmed to convince me to get back on the online platform. But yes, that's my experience so far. We are working. We are work in progress, Prof. Jackson. Thank you.
Thank you so much. Shweta, do we have time for more questions?
No, unfortunately, we do not have time. I believe that everyone is super interested, but I apologize. We are on time. Actually, we just have to finish it off. Thank you so much to the panelists. Susie will be concluding the session now. Just a small word. There are two questions in the general chat room. If our panelists do have some time, we would be extremely grateful if you could answer those questions. But yes, over on to Susie. Thank you so much.
Well, on behalf of the organizers and staff of the Rare Disease Day Symposium 2026, we want to extend our deepest gratitude to Dr. Amar Alkali, Dr. Sasinipa Tritipan, and Dr. Catherine Mutinda. Thank you so much for taking the time to share your invaluable experience and insight during today's fireside chat. Your global perspective on genomic equity have powerfully remind us that advancement in genetic research must be accessible in all rare disease patients, regardless of where they live. We deeply appreciate your dedication to this cause. Thank you so much.
Thank you.
Thank you to everyone who had been attending. You had been a wonderful and amazing audience. We'll now be taking a short 15-minute break. Please help yourselves to some tea and snacks. And during the break, we'll be having a short PowerPoint presentation, which was prepared by our friend Meena. That will be shown on the screen, and it will be showing stories of people living with rare diseases currently around us. And we hope it offers a meaningful perspective as you take a moment to pause in between the symposium. We'll reconvene at sometime past 4:30. We will be giving you an exact time in the chat room. Please do make your way back by then. Thank you so much again.
Welcome back, everyone. We'll now begin the final session of the Rare Disease Symposium. I'm Mashira, and Zarak and I will be chairing this session. Our first speaker of the session is Morgan Devore. Morgan is a PhD candidate at the University of Oklahoma. She will be presenting her work on the Friedreich ataxia. Please join me in welcoming Morgan. Over to you, Morgan.
Hello. Let me share my screen real fast and then we'll do it. 1 second. Okay, can everybody see my screen in full screen?
Yes, it's visible.
Wait 1 second. Sorry about that. Then we'll add a pointer. Awesome. Well, thank you all so very much for allowing me to speak today. I am in the US, so I only got to see the last bit of the last session, but has seemed so great so far. And I just really appreciate being able to share some of my work. So like mentioned, my name is Morgan Devore. I'm from the University of Oklahoma Health Sciences Center, and I'm a PhD candidate. And today we're going to just talk about one of the most recent projects in my dissertation, which has to do with the disease Friedreich's ataxia. So first and foremost, Friedreich's ataxia, or FA, is a genetic condition. It affects about 15,000 people worldwide with a carrier rate of 1 in 100 in susceptible populations. It's characterized by childhood or early onset of progressive loss of coordination and balance, which is ataxia, as well as premature mortality, typically caused by a heart disease called cardiomyopathy. So one of the parts of my dissertation, or I guess my PhD experience that I've just really valued is the opportunity to actually meet quite a few people with FA, as well as their families and the patient advocacy group and so on. So in this picture, you can see my husband and I visiting with Kyle and Emily. Kyle and Emily both have Friedreich's ataxia. And I like to mention just how they describe the disease. So Kyle writes in a book that he wrote that he describes it as almost like this invisible force always pulling him down, while Emily describes it as her battery. Oh, what happened? Sorry, guys. Just 1 second. There we go. Are we back to... Perfect. So Emily describes this as her battery always half full. So it's nice for us in my lab to be able to actually get to know and see these patients with FA, to root our research in helping them and their lives and their families. So, like I mentioned, FA is a genetic condition. Obviously, since we're here today, it's caused by an expanded GAA repeat in intron 1 of the frataxin gene or FXN gene. So patients who have FA have 2 copies of this expanded repeat, one from mom, one from dad, that have over 100 repeats, and very typically they have over 15 or over 1,000 repeats. These repeats cause epigenetic silencing of the frataxin gene, which is used in the mitochondria for ATP production. Sorry. So how we typically diagnose FA is through long-range PCR. So very quick overview, if anyone hasn't been reading these gels, is on these gels, the non-disease causing range will be in this band here, while the disease causing range will be seen within this full range. Non-FA controls will only have 1 band in the non-disease causing range, while carriers will have a non-disease causing and a disease causing allele. And very typically, about 75% of patients will have what we call a double band pattern. So this is the equivalent of saying that one allele has 800 repeats while the other allele has 1,000 repeats. Very typical that you'll see 2 bands in the disease causing range. However, approximately 25% of patients have what we call the single band pattern. So this is typically thought that it's just 800 repeats from mom and from dad, or 1,000 repeats from mom and dad, and that these alleles are just so close that we can't distinguish them on the gel. But due to the large range of disease-causing alleles, as well as just how frequent this single band pattern is, my lab started asking the question, what if we're actually only seeing one allele in these patients on this test, and the other could be either deleted or not being amplified by PCR? So we hypothesized that these patients are actually hemizygous for this repeat expansion. So to test that hypothesis, we used long-read whole genome sequencing. Specifically, we had to use long-read sequencing because with the repeat being over 1,000 triplets on average, short read sequencing cannot go all the way through the repeat, and we had to go through the whole repeat in order to answer our questions. So we used long read sequencing. And the very first thing that we saw was that in 3 out of 25 patients, we found a proximal deletion that encompasses exon 1, the GAA repeat, and the entire frataxin promoter. So as most people here can probably guess, this is a non-functioning allele due to deleting these regions of the gene. These deletions were also independently verified by PCR, by a custom PCR, and they're, I'm sorry, I'm a little bit nervous, and they're consistent with a non-homologous recombination that is done by or through allo repeats. So just to orient anybody to this figure, each line here is one read through the region, and the dark gray boxes are where we see a deletion. So these 3 patients all have this same deletion. FA68 at the very bottom has slightly different breakpoints, but still encompassing the same region of the frataxin gene. So we were really excited to see this at first. Yay, our hypothesis was correct. This person is hemizygous for the GAA repeat. But as I mentioned, we only see this in 3 patients. So, and we tested 25 patients. So we went back to the drawing boards and actually looked at the structure of the GAA repeat in the remaining 22 patients. And we found this really interesting pattern that we're calling a GAA-GGA composite allele. So this is in blue here, this is just one read of this allele. In blue here, it starts as GAA, followed by a long stretch of GGA, and then goes back to GAA afterwards. We were really excited to see this, but just to make sure that this isn't some kind of artifact from sequencing, we looked at all of our patients, and we saw a wide variety of these composite alleles in our 22 patients. So as you can tell, there's a great variety of these alleles, but most of them have some kind of stretch of GGA in the midst of the GAA repeat. So you might be thinking like, okay, so who cares? These people still have the repeat. They're still probably being epigenetically silenced. Who cares? Well, we saw that these composite alleles, they don't quite match the length of what we see on the gel. So for this person, for example, this person, we can see one repeat that's not shown here. That's just pure GAA for 490 repeats. And then we see this composite allele that from sequencing we can tell is about 1,020 repeats. But with the same person, we see on the gel that there's a band at the 490, that's our pure allele, but there's no band at the 1,020 triplet mark. So this led us thinking that the PCR isn't properly amplifying these composite alleles. And we know with PCR that long stretches of high GC content can mess up PCR or make it not amplify that region. But thankfully, we also know a solution to that. So we modified our PCR with the addition of diaza DGTP. And you can see with our standard PCR, this is used by almost all diagnostic labs, about 80% of them. With standard PCR, both of these two patients show as a single band pattern. But with our modified PCR, we can see that both of them turned into a double band patient. So we're thinking, okay, great, we are amplifying our composite alleles here now. But just to make sure that we're amplifying what we think we're amplifying, we used long read sequencing on the products of this PCR. And we sequenced that. So we have many reads of our PCR product. And with our standard PCR, you can see all of these blue lines, they just mean that it's a pure GAA repeat. But with our modified PCR, we can see the composite allele showing up. So we are, in fact, now catching both alleles now with this PCR. We verified with additional patients. So these are our validation cohort of 13 more single band patients. We got about half of our patients having a composite allele, which matches what we saw in the whole genome sequencing. So our method is working, and our frequency of these composite alleles is what we're typically thinking it is. So that's awesome. So we found that approximately 12% of these single band patients have a deletion. Approximately half of the remainder have this composite allele. The other half just had pure alleles, and it truly was just that they had, let's say, 800 repeats for mom and 800 repeats for dad. So it is what is typically hypothesized. But who really cares about this? Well, these FA patients are still being diagnosed as FA. They don't have a band in the non-disease causing range. So they're still being diagnosed. Yeah, our statistics will be a little bit off. We use genotypes of FA to predict things like age of onset and stuff like that. So they'll be a little bit off if we don't catch this composite allele. But nothing too harmful is happening, right? Well, like I mentioned earlier, this is a recessive condition. So there's carriers for these composite alleles that may not be being told that they're carriers for FA. So we next tested families that we know have these composite alleles to see if they're carriers for them. So this is just one representative gel. We've done this in multiple families so far. But you can see with our standard PCR, we have the proband here that looks like your typical FA patient. They have a single band. And then mom looks like a very typical carrier. They have one band in the non-disease causing range, one band in the disease causing range. But dad doesn't look like a carrier. So two things could be happening here. Either dad's not dad, or that dad has one of these composite alleles or a deletion that's causing him not to look like a carrier. We know that he should be a carrier. So we did our modified PCR, and good news is that dad is dad and that he is in fact a carrier of these composite alleles. And now the proband also looks like a typical FA patient with a double band pattern. So I think I went through that really fast, but in conclusion of this part of my dissertation, we have found two novel alleles at the FXN gene that can cause FA. So we found in approximately 3% of all FA patients, they harbor a proximal deletion that takes out exon 1, the full promoter, and the GA repeat, causing this gene to be non-functional, or this allele to be non-functional. And then we found in a substantial amount of FA patients, we have found what we're calling the composite alleles, which are these long tracks of GGA that's not being amplified by our standard PCR. However, we have fixed our PCR, and we are in talks with diagnostic labs even now in order to change the diagnostic paradigm to allow for better genotyping of FA patients, as well as correct identification of carriers with FA. We just recently published this in Human Molecular Genetics. We did a bit more than what I've talked about today. So if anybody's interested in reading more about it, Human Molecular Genetics with the same title as this talk. And then, as always, science is a collaborative space, and all good science has a lot of people involved. So specifically, this is my lab with my mentor, Dr. Bida Chandani, and our lab tech or lab manager, Christina, as well as other PhD student, Lane, as well as funding sources from the Friedreich Ataxia Research Alliance, which is the United States-based patient advocacy group for FA, as well as the United States Department of Defense, where we have gotten funding for this project, Children's Health Foundation, and all of the FA patients and families, and our partners at the Children's Hospital of Philadelphia, who gives us all of our samples. And with that, I know I went really fast through that, so sorry about that. But I would love to take any questions.
Thank you so much, Morgan. That was a very insightful talk. Thanks for sharing your work. We'll now open the floor for questions. You can put it in the chat box or raise your hand.
I will just note one thing. I am legally blind, so it's hard for me to read what's in the chat box. So if you would be willing to read
Yeah,
anything.
no problem. We'll read it out.
Thank you.
Hi, there is one question. I'm going to read it out for you. So does this mean that to positively identify a deletion allele, you must first do a standard PCR followed by your modified PCR, and can only positively call it a deletion if the modified PCR renders a band?
Yes, so I should have been a little bit more clear. I kind of skipped over our deletion PCR a little bit. So the modified PCR that I talked mostly about today is to identify the composite repeats with an FA. So deletion patients are a little bit more tricky. We have done a custom PCR to catch the deletion specifically. So our current workflow is that if a patient comes in and they're a single band patient on our typical PCR or standard PCR, and they're still single band, even on our modified one that shows the composite alleles, then we do a third PCR for the deletion. And the deletion PCR is truly just if there's a band, then there's a deletion. If there's not a band, then there's no deletion. So just a positive, negative PCR that I didn't add into the PowerPoint.
Thank
We
you.
also, to go a little bit more on the deletion, so we've also seen that age of onset and rate of cardiomyopathy and rate of loss of ambulation is severe in people who have these deletions. So that's another clue for us to see if somebody should be tested for a deletion if they look more severe with FA than others and they have a single band.
Thank you so much. There's another question. Can you use triplet prime PCR for FA, or does this have the same limitation as the standard PCR that you mentioned?
Yes, so about 80% of diagnostic labs, at least in the United States and Northern Europe, uses the standard long-range PCR, but triplet primed PCR is also used. With triplet primed PCR, it's typically you've reached over 100 repeats and there's no non-disease causing allele, and that's a good enough answer. But since it's PCR based, again, it's going to have that hard time getting through the composite repeats. So we'll see the same kind of issues with triplet primed PCR, specifically for carriers. So FA will still be properly diagnosed, but not carriers with these composite alleles.
Okay, thank you so much. Mashir, do we have time for more questions?
I think we can take one more question.
Okay. There's one question that asks, are there modifier genes that influence how FA presents?
That is a great question. So I don't know modifier genes. I should definitely know this question or this answer. I don't think modifier genes, but there are downstream effects of like cell pathway downstream effects that can affect FA, the severity of FA. Most of the severity in FA is really caused by the length of the GA repeat. So you'll get much less severe FA if you have less than 500 repeats on one of your alleles. And then typical FA is if you have over 1,000 or over 500 repeats on the other. But there's no just straight up genetic modifier genes for FA.
Okay, thank you.
Thank you so much again.
Thank you so much.
There
Sorry that
are,
that was so quick.
it's okay. I mean, there are other questions as well in the chat box. If you're okay
Yeah.
with it, we'll mail it to you and then you can answer it in your free time, if that's all right.
Yes, perfect. As soon as I
Right.
turn off my camera, I'll get really close to my computer and then answer all of them. Thank you
Okay.
so much.
Hi,
Thank you again for sharing your work. It was very interesting,
Thank you.
especially the modified piece here that you talked about. And now I will hand it over to Zara to continue with the next speaker. Thanks again.
All right. Thank you, Mashira, and thank you, Morgan, for that insightful presentation. Next, I am pleased to introduce Dr. Christy Ross, the Industrial Liaison for the School of Computer Science at the University of St. Andrews. She's a prominent UK Wikimedian and a dedicated advocate for equitable public engagement. She's here to discuss rare diseases and Wikimedia, and please join me in welcoming Dr. Christy Ross.
Hi everyone, and now for something completely different. So I am not going to be presenting any science, obviously, today. I am actually a microbiologist by training, so I've been fascinated listening to all the PCR stuff. It's been throwing me way back. But as Zarek was saying, I'm here to talk about rare diseases in Wikimedia, so collaboration across the Wiki projects. And that's my email address if you wanted to get in contact. Let me just move a few things around just so I can see what I'm doing. Awesome. So let me see if I can navigate through. Yes, excellent. So subtitle for this talk could possibly be Welcome to the Wikiverse. So this presentation, what I'm going to do is I'm going to take you through my journey into the Wiki projects and introduce you to a couple of ways that you might choose to be involved after the symposium. Feel free to get in contact with me if this is something that's of interest to you, as well as a couple of examples of how I've myself, my colleagues, fellow students, and Dr. Saeed Abati have contributed to filling knowledge gaps on the Wiki projects. So and you do need your phones for this one. So if you could get your phones out and do me a favor and scan the QR code out, or you can go to slido.com and enter 797979. I'd like you to pop in a few thoughts of what comes to mind when you think about Wikipedia. And hopefully, she says, hoping the technology works for me, this will mean that we will start seeing a word cloud of things that come to mind when people think about Wikipedia. So if you just spend a minute or two and you literally just start typing, and you can enter multiple answers. It's not presenting it. Why is it not presenting it live? Oh, that's very annoying. There we go. Took a while to update. I'll keep going. Put as many thoughts in as you can. Think first thing that comes to mind. Yay! Someone said everyone can edit it. I'm glad somebody said that. That's good. Okay. Any more for any more? Okay. I might just stop it there, if that's okay, everyone. And we'll go on to, and I'll come back to that later. This is always really interesting. Every time I run this in a presentation, especially with folks who maybe are less familiar with the Wiki projects, I get different answers. Unreliable often comes through, quick answers, general information. Uni doesn't like it. I'm not surprised that that came up. It feeds AI, open source data, don't trust it. Yeah, I'm still having these conversations with folks, even at this point. I mean, Wikipedia's been around for 25 years at this point, so I can understand why people are sort of reticent about using it. I will always say you never cite Wikipedia, you cite the references at the bottom of Wikipedia. Yeah. So first of all, thank you very much for doing that, everyone. I really, really appreciate that. First of all, one thing to say, though, is that Wiki is more than just Wikipedia, and you may or may not recognize some of these logos. These logos represent all the different projects. Don't panic. I'm not going to go through every single one of them one by one. But these logos represent all the different projects associated with the Wikimedia Foundation, which is based in the United States and sort of looks after the wiki projects. There are various chapters. So we've got one in the UK called Wikimedia UK. Big shout out to Sarah Thomas, who's been a great friend and collaborator from there. And yeah, so these are all the different projects, and it's more than just an encyclopedia. You've got things connected to data, to dictionaries, to open access educational resources, to books, to sources, to species, travel guides. This one at the bottom with the 3 triangles, that's an online travel guide that anyone can contribute to. But I'm not going to talk about those today. The main 2 that I'm going to talk about in the context of rare genetic diseases are Wikipedia and Wikimedia Commons. So my journey, I thought might be quite familiar. I'm just going to go through how I found my way into the wiki community and some of the barriers that I found and experienced and how I overcame them. So this was me back in the day, pre-COVID, pre everything like that. My journey started with a wiki editor training session at the University of Edinburgh. This was run by Kandrew. It was brilliant. But as somebody who's neurodivergent, I really struggled with that training session because it was a lot of rules and regulations of how to behave, what are the guiding features of editing Wikipedia. And then when it came to the end of that 2-hour training session, they basically said, right, off you go. And I kind of sat there like a bunny in the headlights going, where do you want me to start? Because even though English is my first language, the prospect of writing and editing Wikipedia was really scary. It was really intimidating. And it actually put me off even getting started. And then, so next step in my journey was the black hole that was, COVID and everything like that, and everyone got locked down. But also, I didn't feel like I could get started. It felt too scary. It felt too big, too intimidating. There was just too many things that I could do it paralyzed me and I couldn't even get started. So Sarah introduced me to Wikimedia Commons and a photo competition called Wiki Loves Monuments. This runs September to October every year in most countries. And it's all about adding photos of listed buildings to Wikimedia Commons, which is a place, if you're looking for good illustrations, audio, visual, that sort of content, that's where it lives on the wiki projects. And the nice thing about that was that it was a binary thing. Is there a photo or isn't there? And so it made it very easy to contribute because it was like, well, if I add a photo, I've automatically made an improvement. Nobody was really judging you. It's technically a photo competition. I kind of treated it like Pokemon Go for grown-ups instead. And yeah, fair warning, that project is a little addictive. I ended up uploading, I think it was 1,500 images last September. Don't worry, I didn't collect them all in September. I did some walks in the summer holidays. This then led me to finding Wikidata, which is, again, It's a binary thing. It's a place for facts, for information, for so for example, Ada Lovelace is the daughter of Lord Byron. All information in Wikidata data is held in triplets like that. It's also a platform that doesn't care what language you edit it in, so it's language agnostic, because underpinning all of it is machine readable numbers, essentially. And then I got a little bit too addictive and decided, what the heck? Let's set up a network at the University of St Andrews called the Idea Network. And that was set up in April 2021. Oh my God, we're coming up on a 5-year anniversary. Very exciting. And so the goal of the Idea Network at St Andrews was to basically make knowledge production inclusive, equitable, diverse and accessible to all people, regardless of their background, location, or culture, ensuring that the wiki platforms and open knowledge platforms reflect the diversity of knowledge in society. And we also try to address the challenge that although anyone can edit Wikipedia, not everybody does. And so far, we've had 53 campaigns with 400 editors. But the big thing that really makes me happy is the fact that our articles that we've improved have been viewed by nearly 3.4 million people. And I think you'd probably struggle to find an academic paper that's been read by 3.4 million people. So it's a fantastic way of getting knowledge out there to readers where it can actually be used. So why does any of this matter? And why should you, in the audience, why should you care about this? Reason you should potentially care is that not all the knowledge can be found on the wiki projects. In English language wiki, for example, an average of about 40,000 people every month edit English language wiki, which if you think in terms of the global context, that's not a lot of people. And those people have a particular interest. They also have a tendency to be white, male, well-educated, live in the global north, have free time, access to the internet. So if you're not one of those things, it is highly likely that your knowledge isn't represented on the wiki projects, which leads to gaps. And these need to be filled by human editors. Fundamentally, at its heart, wiki and the wiki projects are run by people. They are run by people. And it's a community. We argue, we have disagreements, but ultimately, the purpose is knowledge philanthropy, donating knowledge for the greater good. Downstream applications, for AI, that did come up in the questions earlier. They need quality training data. And if your upstream training, if your training data, your wikis, for example, are biased, then you're going to see that in the downstream applications. So for me, that is an argument to get involved with editing these projects, getting involved, contributing knowledge, and making sure that the downstream applications are better by default. So in terms of knowledge gaps, when it comes to rare genetic diseases or even just medical topics in general, the wiki projects around medicine tend to be quite tightly edited. They get a lot of attention because a lot of people rely on them. They're often used as the first answer if you Google something or if you ask Alexa or Siri. They rely heavily on the wiki projects for their answers. And one of the knowledge gaps we found when Saida and I first started looking into this was the fact that if you're looking at the article, say, about pneumonia, really common, condition where you've got a chest infection. If you look at the article in English, it's got about 130 references. It's long. It's got lots of sections, lots of images, treatments, etiology, where it comes from, what causes it, that sort of thing. If you look in Urdu, it's got 3 references and it's about 125 words long. That's a problem. That is a massive problem and also some of those references were in English. So it means if you only speak and read Urdu and the references that this article is relying on are also in English, you can't access them. You can't verify that those facts stated in the article actually appear in the references because you can't read English. And so we decided to focus. So yeah, fixing those articles is really important. But that's where editors such as yourself come in. And also from my little neurodivergent brain, it keeps me happy because there's always more things that we can do. There's always more things that we can contribute to make these projects better. So yeah, the journey never really stops. So what I thought I'd do would be to take you to two examples. So this one in particular, so the Wikipedia one, we were translating articles connected to rare genetic diseases. This has been running for the last 2 years through a project called STEP. Now, STEP is a program, a skills development program that runs during the summer holidays at University of St Andrews, where students can work on projects connected to areas of interest to staff members, in schools or in units, doing useful work, essentially, and gaining skills, building up their graduate attributes. So we kicked off our journey with STEP back in 2024. That's where we started. We then followed it up with STEP in 2025. And in 2025, we actually asked the students to reflect on what the experience was like translating into articles into different languages. We had, I think it was 11 editors last year working in 14 different languages. And the plan is actually to take those student reflections, edit them properly, and get them published on Figshare, so the students then get a publication as a result of being involved in the project. But this is the important one. So they edited 12 articles. They created from scratch 12 brand new articles about rare genetic diseases, edited another 38, adding 61,000 words. This is over the course of 7 weeks, a couple of hours a week for 7 weeks, added 555 references. And those articles have subsequently been viewed by nearly 80,000 people. Once I stop sharing the screen, I will pop the link to that particular dashboard in the chat so you can have a look at some of the articles that they worked on. So that's an example of how you can involve students in translate. And these students weren't experts in the conditions. These were students who often came from modern languages backgrounds or simply just spoke a different language and had an interest. So they had to do a lot of work making sure that it wasn't a straightforward, here's the English article, let's just whack it into Korean or into Mandarin, for example. They had to take the words that were present in potentially in the English, no guarantee that the English article was actually any good. And they had to take that and make it culturally appropriate for the readers in those other wikis. And they also put a lot of effort into finding articles and finding references in particular in the language of the wiki that they were editing in. In fact, one student, for various reasons, ended up hospitalized and ended up peppering her doctors and nurses for questions about these conditions and looking for references, which was brilliant. So that's the content of Wiki. The other thing that we've also worked on is making researchers themselves, so the people who are actually responsible for doing this really important work into these rare genetic diseases, making them more visible. So this might be something that those of you who are on the call could go and do afterwards. You could take a selfie, go and sign up to Wiki and add your selfie to Wikimedia Commons, just as a thought. It then means that when you come to a conference like this, you've got a Creative Commons image that you can use to illustrate your biography, which is cool. But this project in particular was about making sure that when you Google researcher, for example, you're not just getting photos of dead white people like Marie Curie and Albert Einstein. It's actually more reflective of those who are alive and working in research today. So this was a project part of Explorathon, which is Scotland's contribution to European Researchers' Night, runs at the end of September every year. In fact, Glasgow is part of this, so get in contact with Zara Gladman if you're interested in being involved this year. And they basically paid for me to hit the road and drive around Scotland, going to different events. Like this was the Impact Acceleration Festival in Glasgow. I then ended up popping over to the University of Strathclyde to the Department of Chemistry. Dawn Smith over at Napier helped me, and we ended up going around all 3 campuses at Napier University. Stopped off at Heriot Watt to pick up a few more researchers, and then back again to Edinburgh to collect more pictures. And the best thing about this was the fact that none of these researchers had a Creative Commons image up on Wiki. And subsequently, they now have a Wikidata item as well, because most of them have ORCIDs, which is a unique identifier for a researcher. So it makes them more discoverable by other researchers, people they might want to collaborate with, and contributing to the research community on the Wiki project as well. So yeah, so if anyone fancies doing that after this, I would love it to see a whole bunch of photos starting to pop up all across Wiki. So I'm going to stop there and come back to my original question and ask you again what comes to mind. Sorry, this is a very blunt evaluation thing, but it'd be great if you could now maybe pop up a few thoughts about what comes to mind when you think about Wiki and the projects nowadays. And here's hoping things have changed. I've changed a few hearts and minds in this presentation. So you need your phone again. So for the past, and while that's happening, for the past, essentially 5 years, I've been experimenting and collaborating with people outside the University of St Andrews to do all sorts of things, whether it's modern languages, whether it's adding photos of monuments, all sorts of different things. And it can be useful in research, it can be used in teaching. So if anyone on the call is based in the United States and Canada, hi Morgan, in the United States and Canada, there's actually an entire team devoted to embedding this sort of assessment into undergraduate teaching. And also knowledge exchange projects. I'm dying, for example, to run a project where I team up researchers and high school kids who speak the same language to learn to edit Wiki together, to value the fact that speaking in another language is not something to be embarrassed or ashamed of and is actually a real asset. Yeah. So a few folks are still typing, so I'll leave it up for another minute. And I'm very much looking forward to getting into the chat. And for those who are concerned at the beginning that, because anyone can edit it, it's therefore rubbish or unreliable. It depends on how popular the articles are. So if they're in the medical sections, for example, generally speaking, they're very tightly edited. People pay a lot of attention to them because when you edit a page, you can essentially get notified as and when someone else makes an edit. So that's a way of bringing people back into the article to keep an eye on things. It's only really a problem in the sort of the fringes where only 1 or 2 people might be interested in it. And also, for example, we've also got little automated programs called bots that run around Wiki. So if, for example, somebody decided to, I don't know, graffiti a certain politician's Facebook, Wikipedia article, for example, it gets reverted within about 20 seconds. So we've come a long way since it was founded back in 2001, sorry. Okay, thank you very much, everyone. Yay, good to see the changes made. But fundamentally, the best way to learn about the Wiki projects is essentially just to have a go, So please do get in contact with me if you're interested in contributing your knowledge. And there's the email address. So I'll leave that up for a second and happy to take any questions.
All right, thank you so much for the fantastic talk. We'll open the floor for any questions. Anyone can share it in the chat box or they can raise their hand as well.
I know I went through that quite quickly, so that might take a while to settle. Questions are coming in. Let's have a look. Are you not, right, question from Russell. Are you not concerned that AI like ChatGPT will make Wikipedia redundant? And what are the main reasons that people use Wiki? Well, no, I'm not overly concerned that ChatGPT will make it redundant because fundamentally ChatGPT depends on Wiki as a main source of training data and information for it. There's very much the focus is on it being human first on the Wiki projects. AI is probably going to get involved in various things, but predominantly making life easier for the human editors to crack on and get things done. Downsides for things like ChatGPT and other large language models is they often don't tell you where the references are actually coming from. So those references could be coming from Reddit or Facebook, and being presented to you as fact. So the main thing is to work on your critical thinking skills and always question, where did this come from? Who said it? There are various rules for Wiki, for example. It's an encyclopedia first and foremost. It's not a place for original research. Everything has to be backed up by reliable sources. It's got to be written from a neutral point of view, and everything really needs to be verifiable, which means that an awful lot of the references tend to be open access. They tend not to be caught behind a paywall, which is also really, really helpful for people if you want to verify the information that's actually on there. Can we contribute to editing or something? Yeah, you totally can. You can just go up and sign up for an account. You can also edit Wiki with you don't have to use your real name. You can just come up with, a user handle, whatever it happens to be. Mine happens to be Dr. Kirsty Ross because that's my social handle across everything, and it's just easier to remember. You can also edit anonymously as well if you want to, or if you need to for safety reasons, depending on the topics you want to edit. Thank you, Morgan. Science accessibility is so important in today's world. Definitely email me. Excellent. Great. Wiki fundamentally is based on people editing it and changing it and making edits. Sometimes it takes a little bit of convincing to get academics involved, because obviously there are so many things that an academic needs to deal with or do at any one time that Wiki can kind of feel kind of low down the agenda. But it's extremely likely that your work will get read, or your contributions will be read by a lot more people on Wiki and other platforms such as that than your journal articles will ever see the light of day. I'm actually running a research project at the moment investigating where academic research ends up on Wiki, which reminds me I need to actually go and crack on with that project. Is the Wikimedia, after being edited, can translate to other languages useful? Like usual. Not quite sure what you mean by that question. Sorry, Sunny Super. One of the things that we discovered last year, so I was working with a collaborator in modern languages, was, and it comes up quite a lot when you're doing translation work of various different types, is the idea that English is the baseline when it shouldn't necessarily be. It's more that it's a useful starting point. So you can do a straight word-for-word translation, but as I've discovered by collaborating on other projects connected to things like dementia, some languages don't have a word for dementia. So it's how do you write about these medical conditions without the word, without the vocabulary for it? One thing, going back to the person who asked about editing, actually, have a look at Wikidata. Find the genetic condition that you might be studying or working on and see what labels it's available in. Is it available in English? Is it available in Urdu, in Arabic, in German, for example? And just adding little descriptions just makes it easier to find. And it also then ends up on the work lists for editors interested in translating medical articles in that language as well. I literally could talk the hind leg off a donkey about this topic, so I will probably stop there. But do feel free to drop me a line after this if you're interested or want to do any further editing.
Hi, I'm sorry for Dr. Sasinipa's question.
Yeah.
So she's asking if the page can be automatically translated after you have already edited.
It can be. The downside is that, machine translation is getting there, but sometimes it's more work to fix a machine translation than it is just to do it yourself. Reason being, in languages, for example, like Hungarian, Google Translate is actually sexist. So Hungarian doesn't actually have genders when it's talking about job roles. But when you translate from Hungarian into English, it says she the nurse, he the doctor, which isn't great, or she the housewife, he the businessman. So sometimes it does work sometimes, and those translations are getting better, but sometimes it's easier to do it yourself then. And also it's not providing the context, the cultural context that a human would provide with trouble with machine translation. And the other thing you can do, I mean, I've run entire training sessions on this, but you can, for example, connect a genetic condition to other articles by adding in little wiki links, as they're called. So whenever you look at an article, they'll be covered in little blue links, and those link to other places within the wiki. So that can make your articles more discoverable, especially if you're writing them from scratch. But I will, once I stop sharing, drop the link to the dashboard in the chat if anyone wants to explore what the students did last year. I'll stop sharing just now.
I guess we're short on time, so thank you so much, Dr. Ross, for that fantastic talk. So that concludes our session for today. Thank you to all our speakers, chairs, and moderators, and the audience for your engagement. I'll now hand it back to Dishari for the closing remarks.
Yeah, thank you so much, Zarak. I hope my screen is visible to everyone.
Yes, it is.
Yeah. So as we conclude today's symposium, I would like to extend my sincere gratitude to our speakers for their valuable insights, to our alumni and faculty for their continued support, and to everyone who joined us from across the world. Must say the audience was very engaging. We have had amazing questions and we have had great answers from our speakers. Thank you so much, everyone. A very special thank you to Dr. Saida Bhatti. She didn't want to be mentioned, but definitely we all believe we should mention her for her unwavering support and relentless efforts behind the scenes. This symposium would not have been possible without her guidance and dedication. Rare diseases may be individually rare, but together they represent millions of lives. Let us continue to listen, to learn, and to advocate. Before we close, may I please request all speakers, staff, alumni, and students for the group photograph. I request everyone to please turn on their cameras so we can capture a virtual group photo together. Additionally, some of our family members have also joined. I have my parents and Susanna, she also has her parents here. So if all of us could please turn on our cameras for a group photo together. Thank you all once again.